Chromosomes represent genetic material of an organism and are the most stable organic compound that maintains constancy both in number and in structure. However, within populations, chromosomes undergo unusual changes called as aberrations, which involve either the structure or the number of the chromosomes. These aberrations are one of the important biological consequences of human exposure to ionizing radiations and other genotoxic agents. In epidemiological studies, it has been shown that people with elevated frequencies of chromosomal aberrations in their peripheral blood lymphocytes have a significantly elevated risk of developing cancer. Chemicals causing chromosomal aberrations may be identified with an in vitro chromosomal aberration assay. Cell cultures are treated with the test chemical and then mitosis is arrested in metaphase with an inhibitor such as colchicine. The metaphase spreads are examined to detect aberrations. Moving ahead, we now learn types of chromosomal aberrations. Chromosomal aberrations occur when there is a defect in a chromosome or in the arrangement of the genetic material on the chromosome. Very often, chromosome abnormalities give rise to specific physical symptoms. However, the severity of these can vary from individual to individual. Abnormalities can be in the form of additional material which may be attached to a chromosome or where part or a whole chromosome is missing or even in defective formation of a chromosome. Any increase or decrease in chromosomal material interfere with normal development and function. The chromosomal aberrations can occur during meiosis and fertilization and can be categorized into two main types, that are numerical and structural aberrations. Numerical aberrations or variation in chromosome number. Changes in chromosome number may occur spontaneously or can be induced by chemicals or radiations. Changes can occur in whole sets of chromosomes or in individual chromosome. Two main categories of numerical aberrations are euploidy and aneuploidy. Ploidy means unit in Greek, eu means true or even, and anu means uneven. Euploidy. The term euploidy is applied for organisms with chromosomes that are multiples of some basic number n. Euploidy exists in four conditions as follows. Number 1. Haploidy or monoploidy. There are some organisms which has one basic set of chromosomes such as some fungi. In animals, it is not common except in gametes. Note, when whole organisms contains n number of chromosomes, it is called monoploid, and when gametes contains n number due to reduction, it is haploid. Hap means half. Number 2. Triploidy. 3 sets of chromosomes, that is 3n, can originate by the union of monoploid and diploid gametes. These are not common in natural population due to genetically unbalanced gametes, and if present, they are sterile. An interesting case of triploid females has been reported in salamander Embystoma jeffersonianum. In humans, complete triploidy has been found only in aborted embryos. Number 3. Tetraploid. 4 sets of chromosomes, that is 4n, can arise in body cells either by somatic doubling of the chromosome number or by the union of 2 diploid, that is unreduced 2n gametes. Tetraploidy is further divided into autotetraploidy and allotetraploidy. Autotetraploidy. The prefix auto indicates that in this kind of ploidy, only homologous sets of chromosomes are involved. Somatic doubling always produces autotetraploids. Union of unreduced diploid gametes of the same species would also produce autotetraploids. These are most genetically balanced and fertile, but due to various degree of disjunctions, there can be unbalanced gametes which will produce a sterile individual. Allotetraploidy. These are always produced by the union of two unreduced gametes of two different species. Alternatively, somatic hybridization of two different species can also produce allotetraploids. They are generally sterile, but their sterile F1 by chromosome doubling can bring about fertile individuals. Number 4. Polyploidy. Organisms with more than two sets, that is 2n of chromosomes, are known as polyploids. Polyploids are tetraploid that are not encountered in natural populations. Polyploidy in animals is rare and found only in cancerous conditions. In man, cancer cells can have more than 100 chromosomes. Polyploids can be produced by heat or cold shocks or by chemicals like colchicine or echinaptine or by radiations. Aneuploidy. Variation in chromosome number, that is increase or loss, may occur which do not involve the whole sets of chromosomes but only parts of a set. The term aneuploidy refers to this kind of change, and generally this is mostly found in somatic cells, and hence the suffix somic or somi is applied to their classification. Aneuploidy is further divided into monosomy, nullisomy, trisomy and tetrasomy. Number 1. Monosomy. Diploid organisms which are missing one chromosome from a single pair are called monosomic with genomic formula 2n-1. When one chromosome of any other pair is missing, it is called double monosomy with the genomic formula 2n-1-1. Nullisomy. An organism that has lost a whole pair of chromosome is known as nullisomic. It has a genomic formula 2n-2. This is generally lethal. Some polyploids, however, can survive with reduced vigor. Examples of monosomy and nullisomy are also known as hypoploids. In others, where chromosomes are gained, are known as hyperploids. Trisomy. Diploids which have one extra chromosome are represented by the chromosomal formula 2n+1. Trisomy can produce different phenotypes depending upon which chromosome of complement is represented in triplicate. In humans, the presence of one small extra chromosome, that is autosome 21, is responsible for Down syndrome, a kind of mental retardation. Two different chromosomes are in triplicate. Example, 2n+1+1. It is called double trisomy. Tetrasomy. When one chromosome of an otherwise diploid organism is present in quadruplicate, it is known as tetrasomic and is expressed as 2n+2. Structural aberrations or variation in chromosome segments. Structural aberrations involve the change in the structure of chromosome. It also include some disorders which are characterized by chromosomal instability and breakage. One example is the creation of a fragile site on the X chromosome, that is Fragile X syndrome. Structural chromosomal aberrations includes duplication, deletion, translocation, inversion, insertion, isochromosome and ring chromosome. Duplication. It is the phenomenon of having one or more genes in excess of the normal complement due to the presence of an extra chromosome segment. It can be further subdivided into number 1, tandem or repeat duplication. The duplicating segment gets incorporated next to normal corresponding segment. Number 2, reverse tandem duplication. The duplicating segment is incorporated next to normal corresponding segment, but the order is reversed. Number 3, displaced duplication. The duplicating segment is incorporated away from the corresponding segment on the same chromosome. Number 4, transposed duplication. The duplicating segment is incorporated in a non-homologous segment. Deletion. It is the loss or absence of segment of chromosome. The lost segment may be terminal or intercalary. The terms deficiency and deletion are also used interchangeably. Terminal deficiency involves a single break near the chromosome end. In intercalary or interstitial deficiency or deletion, there are two breaks, that is double break, loss of separated segment and the union in the region of breaks. Translocation. It is the transfer of genetic material from one chromosome to another. It can be further categorized into number 1, reciprocal translocation. It is the mutual exchange of segments between two non-homologous chromosomes. In humans, Philadelphia chromosome is produced by reciprocal translocation of a fragment of chromosome 9 into chromosome 22. Number 2, Robertsonian translocation. It occurs when two acrocentric chromosomes fuse to form a metacentric or submetacentric chromosome. This happens due to break in or near centromeres of those chromosomes. This results in reduction of chromosome number. Number 3, simple translocation. A chromosome segment gets inserted in interstitial position of a non-homologous chromosome. Number 4, whole arm translocation or centric fusion reduces the number of chromosomes. Inversion. It is a two-break rearrangement where a chromosome segment having one or more genes gets separated and reinserted in its position, but after inversion or rotation by 180 degree, so that the order of genes is reversed. Inversions can be of two types. Number 1, pericentric inversion. In this kind of inversion, breaks in chromosome occurs on both sides of a centromere and the segment containing centromere gets inverted at 180 degree to rejoin. Number 2, paracentric inversion. When breaks in chromosome occur on the same side of the centromere and only a segment in P or Q region is inverted, this is called paracentric inversion. Insertion. It is when a segment of one chromosome is inserted into another chromosome. Isochromosome. It is loss of one arm of the chromosome while the other arm is duplicated. Ring chromosome. It is formed due to removal of terminal portion of the chromosome, leaving two sticky ends on the central portions that reunite to form a ring. Chromosomal breakage occurs at various stages of mitosis, that is either at G1, S, G2 or M and during meiosis. If break occurs during the G1 stage, only one chromatid is affected and hence it is known as chromatid aberrations. If a short unstained region on one chromatid or misalignment of the proximal and distal parts of a chromatid with an unstained region is observed, it could be classified into gap or break. Chromatid gap include an unstained region not greater than the diameter of a chromatid. The chromatid break on the other end includes an unstained region greater than the diameter of a chromatid. On the contrary, if breakage takes place during S phase, then both the chromatids are affected at identical loci and it is known as chromosomal aberrations. Most chromosome anomalies occur as an accident in the egg or sperm and are therefore not inherited. The anomaly is present in every cell of the body. Some anomalies, however, can happen after conception, resulting in mosaicism, where some cells have the anomaly and some do not. Chromosome anomalies can be inherited from a parent or can be de novo. This is why chromosome studies are often performed on parents when a child is found to have an anomaly. Principle: chromosomal aberrations are the microscopically visible part of a wide spectrum of DNA changes generated by different repair mechanisms of DNA double strand breaks. The inter and intrachromosomal distribution of breakpoints is generally not random. Chromosomal aberration breakpoints occur preferentially in active chromatin. Deviations from expected interchromosomal distributions of breakpoints may result from the arrangement of chromosomes in the interface nucleus and or from different sensitivities of chromosomes with respect to the formation of chromosomal aberrations. Telomeres and interstitial telomere repeat like sequences play an important role in the formation of chromosomal aberrations. Subtelomeric regions are hot spots for the formation of symmetrical exchanges between homologous chromatids, and cryptic aberrations in these regions are associated with human congenital abnormalities. Materials and instruments required: heparinized peripheral blood, reconstituted RPMI 1640 media, fetal bovine serum, phytoheme agglutinin, ethyl methyl sulfolate, colchicine, rectified spirit, hypotonic solution, that is 0.56% KCl, fixative, that is 1:3 acetomethanol, culture tubes, syringes and needles, centrifuge, water bath, incubator, Bunsen burner, micropipettes, microtips, thermometer, slides, etc. Procedure: peripheral blood lymphocyte culture, abbreviated as PBLC, is carried out according to the standard protocol of Hungerford 1965. Whole blood cultures are set by inoculating 0.5 ml blood in 7 ml culture media, that is RPMI media, set at pH 7.4 and 0.1 ml of phytoheme agglutinin. Positive control, that is ethyl methyl sulfonate, is added at a concentration of 240 microgram to induce aberrations. The cultures are incubated for 72 hours. At 69th hour, 20 microliter of colchicine is added to arrest cell division at metaphase stage. The cultures are harvested as per routine protocol and the slides are stained in Giemsa for 10 minutes. The slides are scanned under oil immersion lens for 100 well spread metaphases and are analyzed for chromosomal aberrations. Results: a total of 100 well spreaded metaphase plates are scored for structural and numerical aberrations. Conclusion: in conclusion, it can be said that chromosomal aberrations are microscopically visible changes in the single DNA molecules of chromosomes and chromatids and therefore can be regarded as a kind of visible chemistry. Thank you.